Hey, welcome everybody. This video is being made for my research students, but I'm sure other people could use it as well if they just want to know how to do primer design. So the purpose of this video is to learn how to do primer design based on a gene that you have identified and perhaps a certain sequence in there, and you want to amplify around that sequence. So the first thing that's going to happen, for example, with my research students is, I work with these different mutants and we have 4,000 of them. So for example, LB992. We've identified that our transposon in this case has inserted at nucleotide 1857754, and that's within gene LEPBIA1780. So the first thing we need to do is get the DNA sequence of that gene. So what we're going to do, you're going to get from me basically just this information, 992, insertion site, and the gene ID number itself. So we're going to take that information, copy it, and we're going to go to a website called MAJ. All right, MAJ Genoscope is going to be really, really great for us because it's got all the different leptospira genomes as well as every genome out there for the different prokaryotes you're going to find loaded directly into it. At the top, you're going to see here it's already got loaded in Leptospira interrogans cerebar manilae, but we are not working with that. We're working with Biflexa. So I'm going to put where it says label or gene name, I'm going to copy in that gene ID number just like that. I'm going to click Move To. And when I do that, you're going to notice that now it's switched to Leptospira biflexus, cerebar PATOC1, showing exactly that we're in chromosome 1 in this case. And right here, the highlighted area in this region right here, that's our gene. All right, so we can see here it's LEPBIA1780, the exact one we're looking for. It now also shows directly below it. It shows where the gene locates and resides from the start and finish of it. It also gives information on the type. In this case, it's a conserved hypothetical protein. If you look down a little bit, in addition to that, it also shows you, for example, that it's found and it's got high homology inside of the pathogenic strains we find down here, et cetera, et cetera. So it gives you lots of information right there to begin with. But if you click on the gene itself, if you just do one click, another screen pops up, and this is going to be your object editor. So now you're going to get more information on the gene itself. It shows you start, it shows you end. Again, shows you the product, but if you click up here and if you go down, it's going to give you lots of really, really cool information that can come and play later. Protein ID, the Trumbull sequence, PSORB, where it's located, says the cytoplasmic. So lots of information. This is why we like using Genoscope to begin with. But at the top, your next move you're going to do is you're going to see what looks like a DNA icon. You click on that, and you get the actual sequence for LEPBIA1780. So what we're going to do, we can see on the top, it's the genome sequence, the DNA itself, and on the bottom, this is our protein sequence, right? So we don't want the protein sequence for what we're going to be doing today. We're going to take that gene sequence just like this. We're going to copy it, and we're going to paste it into a Word document. Okay, so we're going to paste like that. It's got LEPBIA there. I'm going to move it down a little bit, and I'm going to put on top of it LB992, so I know that was the mutant itself. I'm also going to go back and find really, really quickly the insertion location. So NT insertion is 1857754. Just like that. I'm going to double check one more time just to make sure. 18577. Yep, it looks like I'm doing a good job so far. All right, good. So I've got this. Now I'm going to put down the start and the begin of the gene so I also know where that's located. So we're going to go back and look. We're going to go through a couple screens here and find our start and finish. We don't even need this anymore. So let's see, start is 1857609. So we can take that information, copy it here because we know that's start, and then we know end is down here, and the end of the sequence is 1858214. I'm going to grab that. And there we go. All right, so now we got start, finish of the gene itself. We know the insertion was at 1857754. If we grab a calculator really quickly, we can find out how many nucleotides in that's located. So we're going to grab the calculator right now, and we're going to say it's 1857754 minus 1857609, and we're going to find it's 145 nucleotides into the sequence. So now what we're doing at this stage is identifying where our transposon inserted. Because remember, we're working with mutants. We created these mutants using, the Mariner transposon elements, so just randomly inserted them into the genome. In this case, it's 145 nucleotides into the sequence. So what I'm going to do is I'm just going to start highlighting. Every line here is 60 nucleotides. So it should be, basically, this should be 120 like that. Somewhere probably right around here might be around 145. So we're going to go up, do some review, do a word count, 143. So if that's 143, 144, 145. It's really just like right in between these two nucleotides here. So we're just going to highlight that, just like that. And that highlight tells us that that's the nucleotide or the transposon insertion site. So now we know where the transposon has inserted. Now for the research we're doing, what we want to do is we want to build primers, all right, for PCR before and after the insertion site, so that way we can identify whether or not the transposon is actually inserted there. So I'll talk about how that is, in class and when we're in the lab, that sort of thing. But what we're really going to want to do at this stage is move up around at least 100 or 150 nucleotides before the insertion site and find our forward primer. So we're going to be finding a forward primer and also a reverse primer. As a reminder, CG pairings are stronger than AT pairings, as we talked about before. That's because CG pairings have 3 hydrogen bonds between them. A's and T's only have 2. So if you want to make a really good, connection between your primer and the DNA itself within the genome, use C's and G's as the start of your sequence and the end of the sequence on the primer, and it will help a little bit. So I'm going to look for CGs. Now, obviously, Leptospira, same as all spirochetes, they're TA rich. They don't have many C's and G's. But if I look right here, you start finding a C sequence here ends with GGG. Let's go ahead and just look at the word count there, and we can see that's 20 nucleotides. 20 nucleotides is great for a primer. What you really want for a primer itself, 15 is okay. It's kind of weak. Really, between 18 to 25 is good. If you do more than 25, what's going to happen is your melting temperature, every nucleotide you add on, the melting temperature of the primer is going to get higher. So it's going to bind so well to the genome, you're going to have to use more temperature basically to break it off. So you could really create a good bond by using more nucleotides, but the more nucleotides you also add on, the higher the cost of your primer. So it's always good to stick between, really I try to do between 18, 24 base pairs. That's a good number, at least in my lab. It works really, really well. You're going to get a good binding. You're going to get strong PCR results. So I like this sequence right here because it starts with some C's, ends with some G's. It's 20 nucleotides in length. So what we're going to do is we're actually going to highlight that as well. And we can highlight these green for the primers itself. I'm going to make a note here, and I'm just going to put down a comment, and I'm going to put down forward primer. So that way I know that's where the forward primer is located. I don't like how it did that. Let me see if I can just reverse that. Let me see if I can take this out. Let's reject that. No. Okay, I know that. Let's see if I can just go like that. First, I'm going to grab it like this, copy it. I'm going to put down here, and in this case, since it's gene 1780, I'm going to put 1780F, and underneath it, there's my forward primer. I'm going to try to grab just the sequence and see if I can make the comment work differently. Good. And I'm going to write down forward primer just like that. So now I know that's my forward primer. Now, again, about 150 base pairs after the transposon insertion sequence, we're going to look again and try to find some C's and G's. And look at this right here. How beautiful. All right. Look at this sequence. Lots of nice C's and G's. It's about 120 pairs later. It's pretty good. Let's see the word count, 21. Love it. Okay, so we're going to now again label this. We're going to give that the green highlighting. And we're going to label this and say it's the reverse primer. So let's see if we go here. Reverse primer. Beautiful. All right. Now, here's the only thing with this, right? So if we put down here 1780R for reverse primer, if I just take this as it is, and I put this down here, and I can do it if I want, it's okay to do that. The only problem is it's actually reading forward, right? So it's actually not reverse. So imagine with this top one, with this forward primer, this is an arrow pointing left to right. Well, down below, as the sequence is reading, if I just take this primer itself, the arrow is also pointing left to right. What I need to do is make the arrow point right to left. So to make that happen, we need to use something called a reverse complement calculator, all right? And so in this case, if you go to Google, and I'm just going to highlight this again so I can grab it, copy it. If you go to Google and just type in RevComp, you'll get this thing to pop up right away. You can see it's right at the top. I just typed in RevComp, reverse complement. Click that right there. Grab your sample sequence. You can write down 1780 so you know what it was. Paste your reverse sequence, your reverse primer that you want to make right there. Click Submit. Boom, gives you this right here. That's the reverse complement. So we can go back here now, and we're going to go back to our document itself and put that sequence in, and I'm going to put next to it RevComp, so that way I know it's the reverse complement. And so when I order, this is the one I want to order. Now we're just going to do a little bit of housekeeping. We're going to do 5' and 3'. We're going to put that in so we know the polarity. Same thing here. We'll put down 5' and also 3'. This also reads 5', 3', so that's good to write down just like that, because that's how it's going to go into the program. And that's perfect, because if you imagine, see how this is like the GGCA? Look at, it's the reverse, it's the complement of this, GGCA, just like that. So you know it's going to actually flip around and attach right there where we need to go and run in the opposite direction. It's going to run upstream for us, right? So this first primer, the forward primer, runs downstream. This primer runs upstream. The last thing we want to know is what the size of our PCR sequence is going to be, like if our PCR product is going to be. So if we do this whole thing, we can write down expected PCR product size. All right, there we go. And let's go ahead and just look here. If we take this right here and we go here, and again, we go up to word count. It's probably around 380. Yeah, 263. Okay, that's all right. That's okay. 263 base pairs. That's okay. That's like the minimum size. 250 is the minimum size you're going to want for a PCR product. If you get smaller than that, it's going to run so quickly through the gel, so it's going to fall off the gel before you actually read it most times. Because remember, we're going to be trying to compare that to actually when there's the transposon insertion. And when you have a transposon in there, it's going to add 3,000 base pairs to the overall size. So 263 is good. Looking at this, I might even say to myself, it's a little wimpy. Maybe I'll pick something down here. I look here, here's a different primer sequence, like maybe just like that, starts with the C, ends with the C. And if I had grabbed this instead, this is probably like 350. Let's see. Yep, 372. I kind of like that better. So I might go back and say to myself, you know what? I'm going to design this primer instead. But this just really shows you how to do the primer design itself. Shows you how to make the forward, the reverse. You would save this document at this point. Save it as LB992 or BIA 1780 primers, and then you can send it to me. And from this information, I can look at what you did, double check that you did everything good, that it looks good. The overall PCR product size is more than 250, ideally between 250 and 400. 500 is fine too for what we want for our purposes. And just double check that you actually did the forward primer, did the reverse complementation, and everything is fine. And from this, I can go then and order primers for us, get those in, and we can do the PCR we want to do. So I hope this was helpful. Short little video, but any questions, you can always email me. And wish you the best of luck with your PCR primer design.